Hi, everybody. I wanted to continue our conversation about vaccines. And remember when the last thing we were talking about was how when you're naturally exposed to a pathogen, that your immune system kicks in, but it does so relatively slowly, and the amount of antibodies you make is relatively low. So, a lot of times, the first time your body is exposed to a pathogen, you actually get sick, but you make those all important memory cells. So once you've gone through this, you make memory cells that circulate in your blood. These are lymphocytes. So B and T lymphocytes that act as memory cells. And so the next time you're exposed to something, the same pathogen, you're going to make a much quicker immune response and it's going to be much stronger. So a lot of times you won't get sick twice. Like a lot of people that have had chicken pox never get it again, something like that. Um, and so we have the primary and the secondary immune response. Now, what this has to do with vaccines is a vaccine can be used to give you a primary immune response instead of a natural pathogen, and that will allow you to make memory cells and prevent yourself from getting sick when you actually are exposed to the pathogen in nature. So, if you look at the four most common kinds of vaccines, the four kind of vaccines that we've had for quite a while, they all kind of, the role of these vaccines is to stimulate a primary immune response and to get your body to make memory cells. And you can see from this table that there's a nice description, advantages and disadvantages. This table, I believe, is in your book. So I have four different kinds of vaccines here. And the first one is live attenuated. Now, attenuated means weakened. So what's happening when you make a live attenuated vaccine is it says a weakened strain of a whole pathogen. So what can happen? Here, give you some examples. The chickenpox vaccine, the measles vaccine, lots of our common vaccines that we get are live attenuated. So it's actually, in all these cases, these are viruses and they're able to infect us, but they've been weakened in a way that when they do infect us, they don't actually make us sick, but they do stimulate our immune system to be able to make antibodies and make those memory cells. So here it says advantages of them is we get a good amount of immunity. It's long lasting, and that's actually important because this virus is actually still alive. It can continue to stimulate your immune system, so it generally gives really good immunity. Disadvantages, one of the big disadvantages is if this is live, it says there's a risk of infection to immunocompromised patients. So for most, for healthy people, the vaccine won't have any negative effects on them as far as causing disease. But often with immunocompromised people that have a weak immune system, even this weakened virus may be able to cause some disease. So that's something to be aware of. The other one that can happen is there's some kind of mutation that the virus mutates back into a more pathogenic or more one that's more able to make you sick. So again, these are rare events, but they are possible because it's a live virus. The second of the four common types of vaccine, this one says inactivated. So instead of putting a live but weakened virus in the patient as the vaccine, this is actually an inactivated virus. It's what we would, if it were a living organism, we would say it's dead. It's not able to infect cells. It's not able to replicate, but your immune system does recognize that it is foreign, and you can make an immune response against it. So it says the whole pathogen's killed or inactivated, and it gives you some examples of how they do that. A little bit easier to store because it's not alive. generally you'll get a weaker immunity from this kind of vaccine. And there are some examples of the kinds of vaccines that we do this with. Now, one of the things you think about as we're looking at these four types of vaccines is what determines which kind of vaccine we use, and on these various examples. And one of the things that, well, just kind of the main thing is you want to find a vaccine that gives you the best immune defense as possible with the fewest side effects. And particularly if you're like a really dangerous disease, you don't want to take any risk that you might get something that reverts. So when you get to some of these things like anthrax, hepatitis, meningitis, some of these things that can be quite serious, using a live vaccine might not be as good of an idea. Okay. Subunit vaccine. So it is what it says it is. It's a subunit of the virus or the bacterium. And remember when we looked at, in fact, we can go back and look at, oh, this picture has it here. Here's some bacteria, a bacterial cell, and it has different proteins. And these proteins are the antigens on the surface of the bacteria. And viruses also have unique proteins or unique antigens. And so what the subunit vaccine is just molecules of these antigens. They're not the whole organism, the whole cell, or the whole virion. Okay, they're just proteins or antigens. So here they say the subunit vaccine is just the antigens, and it says immunogenic antigens, which means antigens that can stimulate the immune system. Lower risk of side effects, but what it says, limited longevity, multiple doses required, and but these are vaccines that we use, and there's a lot of that we've all been vaccinated against. So I used to hear here it says that influenza vaccines. Oh, influenza vaccines are made a couple different ways, but you can see that they're inactivated or they're subunits. And I remember a student of mine telling me she worked at a nursing home, and she would say, "Oh, when everybody gets their flu vaccine, then everybody gets a mild case of the flu afterwards." And I thought that didn't really make any sense because the flu vaccines are either from inactivated or subunits in the vaccine. So these don't multiply, they don't cause the signs and symptoms of influenza. What does happen when we get vaccines quite often, and a lot of us experience this with our COVID vaccines, is just that our immune system kicking in and recognizing there's pathogens in our bodies or something that looks like a pathogen. And a lot of times you'll get body aches and fever and stuff like that as a result of our immune system response. So it's not necessarily those side effects that you might get from an influenza vaccine are not the virus causing a mild case of influenza. It's your immune system responding to that virus and causing some of the same signs and symptoms. Finally, the fourth major type of vaccine are toxoid vaccines. And remember, we had the words when we had phenol and phenolics. We said phenolics are variations on the molecule phenol. And so toxoids are actually variations, created in the laboratory of toxins that are made by bacteria. Now, we haven't talked too much about bacterial toxins yet. We're going to talk about that in the next unit. But some bacteria actually, when they infect our bodies, it's not the bacteria that make us sick, it's the toxin. And so what they do is they inactivate the toxin so it doesn't make you sick. But you are able to produce an immune response to those toxins. So here it says it causes this immune response. The disadvantage, though, is they don't stop the bacteria from infecting you. They just stop the toxin that the bacteria makes from making you sick. And really the one I think about the most is tetanus. We all get tetanus vaccines. And the tetanus bacteria, remember it's Clostridium, the genus Clostridium, Clostridium tetani. The tetanus bacteria itself doesn't make us sick. It's the toxin that's produced by the bacteria that causes us to have that, causes your muscles to stay contracted and not relaxed. So this is a toxoid vaccine. So these four vaccines, these four groups of vaccines are the four major classes of vaccines. So make sure you know what is in there. So this description is the main thing you want to know. There is some little practice in your lecture notes. So why don't you try that? You can stop this video right now and try it right now, or try it when the video is done. But we can talk about that on Monday in class. On your lecture guide that I gave you, your printed objectives that you have, I actually forgot or neglected to add this section. So you're going to add this in there. That I just wanted to also talk a little bit about some of the new style vaccines that we've been making as a result of the SARS-CoV-2 or COVID-19 pandemic. And so add in there that I wanted you to understand a little bit about these newer vaccines. And so the fifth kind of vaccine we have, and in addition to the four we just talked about, are mRNA vaccines. So remember when we were talking about the central dogma, we said DNA is used to make RNA, which is used to make proteins. And I think most of you knew that DNA is used to make messenger RNA, mRNA, to make protein. So it's a special kind of RNA. And both the Pfizer and the Moderna vaccines are mRNA vaccines. And this picture gives you an idea of what's happening there. And here they have a piece of mRNA. So we have DNA to RNA to protein. Okay, they get a piece of RNA from the COVID-19 virus. And that RNA, the piece that they get, contains enough information to encode a protein. So it's a copy of one gene. And that one gene is the gene that makes the spikes on the outside of the virus. Okay, remember that coronaviruses have all those spikes that look like a crown. And so the RNA that's in the vaccine is the SARS-CoV-2 RNA, enough RNA to encode for the spike protein. And what they do is they package that RNA into a little, I've heard it called like a lipid bubble, like a little vesicle kind of, like a little lipid bubble. And so when you get an injection, what happens is those lipid bubbles go into your muscle in your arm, and they go into your, they can even get into your lymph nodes in the area. And so this would be like a muscle cell or an immune cell that this mRNA gets in. So here's that lipid bubble. And remember that our cell membranes are also lipid. So this will fuse with the cell membranes just in the region where you got the injection. And that mRNA is released in the cell. Here we go. Translation, that RNA hooks up with the ribosome, and now you make a protein, an amino acid chain that makes a protein. And the particular protein you're going to make are these spike proteins from the COVID-19, SARS-CoV-2 virus. Okay, and then what happens is when your cell makes these proteins, it actually transports them and puts them on the outside of the cell. and that allows your antibodies to go, oh, there's this protein that's foreign to my cells. We're going to make a lot of antibodies against there. And not only are you going to make antibodies, but you're going to make memory cells. Okay. So the questions that people have about this a lot of times, there was this big thing like, oh, this vaccine changes your DNA. It doesn't even get into the mRNA doesn't even get into the nucleus, and it doesn't interact with our DNA at all. And you guys learned earlier this semester that RNA is not very stable. So even just a few hours after after this RNA is injected, or I think it's actually more like a few days. After a few days, this mRNA is completely gone. mRNA is not very stable. So you're going to make a lot of protein, and then the RNA is going to degrade and it's gone. Okay. The other thing, here you're going to make proteins. The proteins are going to stay around for a couple weeks at the most. And they're going to stimulate the immune system. And so they eventually kind of deteriorate too. Okay, so the mRNA vaccines, the vaccine is actually a piece of mRNA that encodes for the virus spike protein, and it's in just a little lipid bubble. Okay, that's our mRNA vaccines. The other kind of vaccine that you've heard about with the SARS-CoV-2 pandemic, it was the Johnson & Johnson vaccine. And that one is a viral vector vaccine. And, it was kind of interesting when they were just starting all this, trying to make these vaccines, and there were a whole bunch of different types they were working on. In fact, they were even using three of the four conventional types that we talked about on the earlier slide. So, in fact, let's look back there. People were working on making live attenuated vaccines against SARS-CoV-2, inactivated subunit vaccines. We weren't making toxoid vaccines because this virus doesn't make a toxin. But it turned out these more modern vaccines were just a lot faster to make, and they worked. So the viral vector vaccine, so what you do to make that is there's actually, you do kind of what you did with the previous vaccine is you get a gene to encode for that spike protein. And so here's the virus. Let's say this is a SARS-CoV-2 virus. They end up taking the RNA out, and you end up isolating a gene for the spike protein. And they put that inside, instead of using the SARS-CoV-2 virus, they actually put it inside of kind of a dummy virus. This is the vector part. The vector is kind of, it's actually an adenovirus, and these are really common viruses in nature, in humans, and they have one that doesn't cause any kind of disease. And so what they do is they take out some of the genes out of that, and they put in this SARS-CoV-2 gene. So it says this adenovirus is used as a Trojan horse to deliver genetic code into cells. So it's going to deliver some of this SARS-CoV-2 spike protein gene into it. And so when our viral vector vaccines made out of this, you get an injection of this, and it actually does infect your cell, and it causes the viral proteins to be made, and in a similar way to the previous one. And those viral proteins will do the same thing as the previous slide. They stimulate your immune system to make antibodies and memory cells. So it's a little bit different mechanism, but again, it seems to be working pretty well. Has some advantages as far as stability, but this is one of the vaccines some of you probably already got. Okay, so those are the types of vaccines. And the other thing, you guys, I think most people have gotten really good at understanding the concept of herd immunity. You might have heard this called, I think they tried at one point to get people to start using the terminology community immunity because it maybe was a little more descriptive. But herd immunity seems to be the term that's more popular. And so this diagram, I don't think this is in your book. I don't think it is, but it's really easy to find it on the internet. Just Google herd immunity and you can find this. But what they're trying to show you, actually, let me just give you a definition of herd immunity before I go on. Herd immunity occurs when enough people in a population are immunized against a pathogen to confer protection to the few people who are not immunized. So herd immunity is when enough people in a population are immunized or protected from a pathogen to the point that they are able to protect the few people that are not vaccinated or not immunized from that pathogen. So let's look at this diagram. This one right here, these blue guys, these are individuals are not immunized, but they're healthy. And what most of you can see, I don't know if the colorblind folks can tell this one, but here's a red guy, here's a red guy, and all the rest are blue. The red ones are, they're not immunized and they are sick and contagious. Okay, so the blue's not immunized but healthy. The red's not immunized. I'm sorry, the blue's not immunized but healthy, and the red is not immunized but sick. So what's going to happen in that case? These red people are going to they're going to infect the surrounding people who are going to infect the surrounding people, and most of the people in that population would end up getting sick. So a contagious disease is going to spread through that population. If we have some of the population immunized, and right here, this yellow guy in the middle is immunized and healthy. So we've got the blue people that aren't immunized, and that makes up most of this picture. We've got these two red folks that are sick, but we've got a scattering of yellows, and these yellow individuals have been vaccinated and are not going to get sick. So what happens? We still get a lot of people sick. Most of those blue people still got sick, and the ones that were protected were just the people that were immunized. Okay. So if we have a small rate of immunization, the only people that are being protected are those people that are immunized. So now the idea is, how do we get herd immunity? So again, here we have our two sick people, but now we have most of our population is immunized. And we just have a few individuals in that population who are not immunized. So these blue ones are not immunized. Most of them are yellow. They're immunized. And then I've got these two red people that are sick. And what's going to happen? These red ones can spread that disease, but look they were pretty much surrounded mostly by people that have been immunized. So you pass it to somebody who's been immunized, they don't get sick, they don't pass it to anyone else. And what can happen is some of these folks that were not immunized are actually protected from getting sick. And that's what we call herd immunity. Now, it doesn't mean everyone's protected. Here's somebody that got sick anyway. But herd immunity should help us prevent people a small number of people who are not immunized from getting sick. And who are those people? Well, they should be people like infants who have not been able to get vaccines yet, infants and young children. They might be immunocompromised people, people with a naturally low immunity. Like I think I mentioned, I knew somebody who had a child that had an immunosuppressive, was born that way, had a suppressed immune system. Um, People on chemotherapy whose immune systems are depressed because of the chemotherapy. So there's all sorts of reasons why somebody might not be able to get immunized or might be sensitive, might be more likely to get, might have low immunity. So herd immunity is designed to protect some of those individuals who are not be able to be protected otherwise by vaccines. This table is kind of interesting. It it what it's showing you is this number called R naught. Um, it's a measurement of how contagious something is, and it actually allows you to calculate what percent of your population needs to be immunized to provide herd immunity. And the higher the R naught, the easier the disease spreads. So what this means is if somebody has mumps, the R naught is 4 to 7. And what that means is a person with mumps is likely to spread that disease to between 4 and 7 other individuals. Somebody who has measles is likely to spread that disease to 12 to 18 other individuals. And so what we're saying is measles is a lot more contagious than mumps. And there's a calculation, there's a formula that you can use to calculate what percent of people need to be vaccinated in order to get herd immunity. And what you can see here is with mumps, if 75 to 86% of your population is vaccinated, you'll be providing herd immunity. You'll be protecting those individuals, the few individuals who are not vaccinated. Measles, because it's so much more contagious, you need to have 83 to 94% of herd immunity of people vaccinated to get herd immunity. So it's going to depend on the pathogen as to what percent you need vaccinated. But probably a good number to remember is 80%. If you can get, just a general number, if you can get 80% of your population vaccinated, in many cases, you're going to be able to get herd immunity. Now, you hear this pertussis, which is whooping cough. You need more than that. But just to get kind of a guide, you're somewhere around 80%. And so the numbers that I found, and I think that the CDC is using something similar to this, the COVID-19 R naught is estimated to be about 2 to 3, so it's pretty low, making herd immunity about 60%. But I've actually seen this number that I've heard 80% being recommended. But I think it depends on the population and how close people are living and that kind of thing. But something like that, so we want to shoot for like 60 to 80% for COVID-19. Anyway, that's all I'm going to talk about with viruses, with vaccines. Okay. So how do vaccines work, our 4 major kinds of vaccines, plus our 2 newer kinds, and just a little bit about herd immunity. I think I'll give you a little bit more on antibiotics so we don't have so much to cover on Monday. But let's talk a little bit about antibiotics. The objective says, describe the approach being taken by Paul Ehrlich to control microbial growth and his concept of the magic bullet. Turns out there were, we had the golden age of microbiology. And that's when people figured out that the germ theory of disease. And then lots of people started doing research on trying to figure what pathogens are going to cause what disease. And you can imagine the next step is going to be, wow, now that we know the pathogen, if we can kill the pathogen, we'll be able to treat the disease. But the trick that you have to realize is that there's lots of things that can kill bacteria. We just spent, a few days a lecture on that. There's lots of things that kill bacteria. But the problem is a lot of those are also going to kill the patient, like us. And so the trick was to find what Paul Ehrlich called the magic bullet. And so what that means is, think of a magic bullet that you can shoot somebody with and it kills the pathogens inside of them, but it doesn't kill them. Okay, so it's a magic bullet. He's looking for a magic bullet to kill pathogens without hurting the host. And so prior to 1940, Civil War, World War I, there were all sorts of chemicals that were used to try to treat infections. Iodine, carbolic acid, hydrogen peroxide, all sorts of things. And some of these things actually were so toxic that in retrospect, a lot of times people think it's the treatment that killed the patient more than the pathogens. So lots of these things were really toxic. But Paul Ehrlich actually got credited with creating the first actual drug, the first magic bullet that targeted a particular disease and worked effectively. And so what he was doing, he was working in his laboratory with all sorts of things that we know kill bacteria, things like arsenic, nasty chemicals like that. But he did some modifications of some nasty chemicals and came up with something called salvarsan. And salvarsan was actually used to treat syphilis. And so it was our very first drug that was used to treat a certain kind of pathogen. So Paul Ehrlich gets credit for coming up with the first magic bullet. High toxicity was a major problem for this drug and many of the chemicals used at the time. But what happened in 1928? Good old Alexander Fleming saw that mold and discovered that mold kind of inhibiting the growth of bacteria, and we came up with penicillin. So antibiotics became around. We started using them in the 40s around World War II. In the 50s, Selman Waxman, who this is, he won a Nobel Prize for the discovery of another antibiotic called streptomycin. And he actually ended up discovering about 20 antibiotics. And he found them in soil. And, there's lots of bacteria in soil. There's lots of competition there. And so he was able to find them. And they're easy to find. Any of us can get bacteria and soil samples and find antibiotics. So he actually came up with the name antibiotic to describe an antimicrobial agent that's produced naturally by an organism. Today, the word antibiotic is an antimicrobial from a natural, semi-synthetic, meaning you got something natural and fiddled with it in the lab, or completely synthetic in origin. So antibiotics can be all sorts of things. We might have even called salvarsan an antibiotic if it came later on, just a synthetic one. Okay, I think I'm going to stop right there. And we can pick up right here and talk about how do antibiotics work and go with the rest when we come back to class on Monday. Okay, so save your questions for me. I'll be glad to answer them or send me an email. You guys take care.